Screening by functional complementation. Complementation, it is a process in which we can compensate the missing function of a particular cell or bacteria so that the wild type phenotype it is restored. And this process where we are going to restore the wild type phenotype, it is called as functional complementation. And this process it has also been used to screen specific target clones. And this procedure of functional complementation was used in late 1990s by different workers. For example, first time functional complementation was used by using eukaryotic DNA, where it has complemented the function of Auxotrophic E. coli. Now, auxotrophic means that it is a process in which a particular function of the gene it is lost, so that the cell is no longer able to prepare particular organic molecule which is required for its normal growth. So it is also called as auxotrophy. So auxotrophy हम उस सिचुएशन को कह सकते हैं जहाँ कोई ऑर्गेनिज्म जो है किसी खास ऑर्गेनिक कंपाउंड को प्रोड्यूस नहीं कर पाता, और ये जो ऑर्गेनिक कंपाउंड है ये इसकी ग्रोथ के लिए ज़रूरी होता है. तो जहाँ जो फर्स्ट टाइम कॉम्प्लीमेंटेशन प्रोसेस को यूज़ किया गया, जैसे मैंने कहा 1990s में, तो इसमें एक ऐसा E. coli auxotroph यूज़ किया गया that was unable to produce amino acid which is like histidine. So E. coli mutants were not able to produce histidine. So by using sequences from the eukaryotes like the yeast that produce histidine molecule, they were used to complement the function of that gene in E. coli. So the library was constructed by using different clones from the yeast cell that also contain the gene for histidine biosynthesis, and then the recombinant molecule or vector that contain histidine biosynthesis gene, it was used to transform Auxotrophic E. coli. When it was introduced, then the screening was carried out that which cell contain the recombinant molecule and have the ability to grow on a minimal medium, because E. coli cells can then only be able to grow on a minimal medium when it has the ability to produce histidine. So this is one of the first example where functional complementation was used to restore the wild type phenotype in case of his B mutant E. coli, so that now it is able to produce histidine in the presence of recombinant molecules that contain histidine biosynthesis gene from yeast cell. In addition to microorganisms like the E. coli, functional complementation it can also be used to restore lost functions in case of higher organisms like plants or animals. Again, I can quote one of the example where the gene that mediate the process of deafness in mice, like the Shaker2 gene, it was replaced or restored by functional complementation. So Shaker2 gene, it control the deafness in mice, and here the wild type genotype it was constructed by using bacterial artificial chromosomes, and then it was injected in the eggs of mice that contain Shaker2 mutations that control the deafness, so that the normal phenotype which contain normal hearing it is restored. This diagram it indicated the process of complementation to restore the Shaker2 mutations in mice. Now what we can do in this process, that from the wild type mouse that have normal hearing, the genomic DNA, it can be extracted and then it can be digested with appropriate restriction enzyme, and then it can be ligated with bacterial artificial chromosomes, and then it can be further screened for back clones that contain the wild type phenotype that can be restored in mutant mice. So here the back clones, it can be injected with homozygous Shaker2X that are mutated, and after injection then the mice can be grown from these eggs, and then the presence of normal hearing, it can be screened, and here it is the normal mice, and then it can be subjected to further screening later on. So these are the different screening strategies that can be used to screen either genomic or cDNA libraries and even their expression products. So overall, we have discussed that screening, it may be on the basis of the presence of vectors or even the hybridization techniques that may be sequence dependent. They can be used by using specific primers or the probes in case of hybridization dependent screening. In addition to that, the screening may be based on chromosome walking, chromosome jumping, and later on we discussed that how the expression libraries can be screened by using immunological methods, southwestern or northwestern method, and how we can perform functional complementation.